Good afternoon and welcome to today's EMSL LEARN webinar, everyone, where today we are going to be talking about visualizing live cell dynamics and metabolic activity through chemical probes. The EMSL LEARN webinar series is designed to provide information to the scientific community on resources available at EMSL for research into the biological, environmental, and computing sciences. Our webinars are posted on the EMSL LEARN section of the EMSL website, so we invite you to visit that section to see past webinars. If you want to stay informed on upcoming webinars and upcoming proposal calls, you can sign up to receive emails from EMSL, or you can go ahead and follow EMSL on social media, and the links will be posted in the chat. So for today, our presenters include Sankar Krishnamurthy, an EMSL chemist who will discuss how you can use activity-based probes to gain a spatial temporal understanding of functional proteins in biochemical processes. Kevin Crampton, also an EMSL chemist, will discuss how coherent Raman spectromicroscopy can be used with activity-based probes to visualize transport and metabolism in live cells. And lastly, but not least, Damon Leach, who you may remember from the last webinar that we had. He is a PNNL data scientist who will show you how to get the most out of your chemical probe data with ChemProt-R. If you have questions during any of the presentations, please post them in the Q&A section, which you will see just at the bottom of your Zoom screen. We will answer them during the Q&A session starting roughly around 12:45. We will not be answering them directly in any of the chats, so please know that we will answer your questions at the end. And with that, today we will start with Sankar.
Everyone, once again, I'm Sankar Krishnamurthy. I'm a staff scientist here at PNNL. My background is in chemistry and chemical biology, and I also steward the chemical biology research area for EMSL. Today, I'm going to introduce you to chemical probes, what they are, how we make them, how you can use them for your research. My goal today is to give you enough background so that you can suggest chemical probes as one of the analytical tools that you can use for your current research and also for your future proposals. What are chemical probes? These are small organic compounds with a molecular weight of anywhere between 200 to 1500 Dalton, and that we use them to disrupt the biomolecules to answer mechanistic and phenotypic questions. I have listed here 3 types of chemical probes. The first one is classical modulators. These are either naturally available or synthetically made in the labs that you can use them to disrupt the biomolecules that results in observable phenotypes. You can use that to study genetic and environmental differences. And the next, we have activity-based chemical probes. These are pioneered by Ben Crowat at the Scripps Research Institute and widely used by the chemical biology community. These are designed in a way that they are only activated by the active enzyme that it is targeting. When it is activated, it irreversibly binds the target enzyme. And the probe has a reporter group, which then can be modified to further understand this protein. The next, we have activity-based fluorogenic or Raman probes. These are probes that are very similar to the activity-based probes, except that they do not irreversibly bind. Instead, they release a chemical species that has a altered fluorescence and Raman signature that you can use the respective spectroscopy to characterize the enzyme activity. I'll be focusing on these 2 types of probes today, and among them, they can give you insight into some of the things that I have listed here. So now that I introduce you to the chemical probes, let's take a closer look at the probes and their structural features and how they are made. So typically, the activity-based probe has three element: a reactive element, a recognition element, and a reporter element. The reactive element allows the probe to irreversibly bind the protein target. The recognition element is how the protein recognizes the probe as its own ligand or a metabolite that interacts with, or a substrate that it turns over. And that actually changes significantly depending on the biology and the biochemistry that you're looking at. And we have a reporter group that are typically a modular chemical functional group that you can modify using click chemistry to perform downstream analysis. So when we are looking at enzyme, there are hundreds and thousands of enzymes out there. It's difficult to make a true activity-based probes for every single enzyme. So the community developed what is called a photo affinity probes. These are compounds that get activated in the presence of light, where the reactive group is a photosensitive group. When you expose it to light, it forms a reactive group that allows the probe to irreversibly bind the protein that is at close proximity to. So how do we make these probes? So we need a lot of background information to successfully develop a probe. So we rely on past literature to design these probes. First, we look at, do we know biochemical information? Do we know what enzyme it targets? What is the substrate? We take account of those information and design a probe that is synthetically tractable in the lab. And we go out there and using organic chemistry methods, we develop and synthesize and characterize those probes. Once we have the compound in hand, we go forward and perform further validation to see if the probe or the compound actually targets the enzyme that we're saying it targets. For that, we use either commercially available pure enzyme or an organism that has the target enzyme to characterize how the probe performs. So this allows us to understand the limitations of this probe, so that when we actually go forward and apply in the biological system, we understand what are the limitations of this probe. Once we characterize that and we consider that's validated and it is ready for use in the actual system we want to use. So with a probe in hand, you can either use it in a proteome that is extracted from your organism, or use it in a living cell directly. You take the probe, incubate it with your sample, and the probe gets attached to the target enzyme, which then can be coupled to biotin using a copper catalyzed click chemistry. That allows us to enrich for the targeted proteins by streptavidin beads. The beads then digested by trypsin, and we get peptides. Those peptides are analyzed by mass spectrometry coupled to liquid chromatography. You get the mass spectra of those peptides, which then rolled up to call the protein targets that the probe identified. Third portion of this webinar, Damon will go through the process behind how the peptides are used to roll up to call the proteins. So you can also couple the probe labeled proteome with fluorophore, where those labeled proteins then can be developed into the SDS page gels that are separated based on the protein's molecular weight. And we can also look at the probe labeled whole cells under a fluorescent microscope to give you additional information. And if you're interested in enriching for a certain population that is labeled by the probes, we can use fluorescent activated cell sorting to enrich those probe labeled cells, which then can be used for further analysis. So this is the current activity-based protein profiling workflow that we're currently using at EMSL. This gives you what kind of pipeline your sample will go through. I'm gonna give you a few more examples in the next few slides on different types of probes. The first one is a probe that specifically target one particular enzyme. And so I should step back and orient you the slide. You'll typically, in the next few slides, you'll see the substrate and the probes on this side, and additional information about the probe targets and organism, and who are the end users at the top. And any other additional information that I want to give you or show you data will be here. And then you'll see the contributing researchers at the bottom. All right, so this probe specifically designed to target an enzyme called C5a peptidase. We knew that the enzyme breaks down a protein called C5a at a particular peptide sequence, and we use that information to modify that amino acid into a probe, which is already coupled to a fluorophore. After certain validation, we use this probe to show that if you had a mixture of the target organism and a non-target organism, you can actually separate using fluorescent activated cell sorting. So that's an example for a specific enzyme. The next example where we know what the metabolite is, but we want to see how this metabolite is transported and moved in the fungal system. So we modified the sugar into a photoaffinity probe, where we have this photoreactive group and a fluorophore. Once the probe is ready, we incubated with the fungus Yerevia lipolytica, and we exposed the whole probe label cells to light to activate the probe so that it irreversibly binds its target. And we washed off the cells and took this fluorescent imaging. You can see that the fluorescence dose dependently increases. After that, we lysed the cells, extracted the membrane fraction, and ran STS-based gel, where we see there's a labeling around the 50 kilodalton molecular weight region. That was promising, so we moved forward and used the activity-based protein profiling and analyzed the sample by mass spectrometry. In that analysis, we found two proteins labeled as transporters. So that gives you an idea how you can go about narrowing your sample so that you can gain information about the targets that you're looking for. And the next example is pretty similar, where we're taking a sugar alcohol called myoinositol, which we're modifying it with the similar photoaffinity linker and a reporter group. But in this case, the target organism is different. We took this poplar plant and we performed about 6 injections of this probe with a 2 days interval between the injections. And then the root was exposed to UV light to activate the probe. And we harvested the root and extracted the proteome. You can see that in the STS-based gel, we see a few bands that are fluorescent, which then we excised and extracted the peptides from those gels and analyzed by mass spectrometry. With that analysis, we found 4 different transporters, 4 different targets labeled as transporters. And we're actually hoping that we can use this probe to further perform the actual activity-based protein profiling. And this probe and this method is available for users in the upcoming calls. The fourth example is very similar, but except that we're not using sugars anymore. Here, these are terpenes. This is part of a user project that was awarded to a Hamburger lab at the Michigan State University, where they're interested in understanding the communication between sorghum root and the root-associated microbiome through the terpenes. So they are interested in having a few of these terpenes modified into probe, so they can track how who's taking up, which microbes are taking up these terpenes, and what are their protein targets in their microbe. So far we have made 3 different compounds, and then in their new project we're making 2 more probes. This is an ongoing project where we'll be using all these 5 probes to see which microbes are taking up these probes using fluorescent activated cell sorting. We'll also perform the activity-based protein profiling to identify their protein targets in those microbes. The next example is a little bit different from the ones that you've seen so far. These involves the second types of probe that I showed you, where these are just turn on type of probe, where the enzyme upon activation releases the fluorophore. But here you can imagine a scenario where you do not know what the target enzyme is, and you do not know what the substrate of those enzymes are. So you only have an idea of a biochemistry or a biochemical function. One good example is that the soil bacteria, for example, consumes chitin from soil, where people understand that it takes up this NAG and turns it into glucosamine and used up as a nitrogen and carbon source. So the first step is basically the amide hydrolysis of these amide. So the probe that we developed, the probe library that we developed, is based on the idea of having some sort of amide hydrolase function, right? So we developed this library that had a variety of carboxylic acid coupled with a fluorophore. So this R group here has a variety of structural features, and we have 1 fluorophore. And so you can imagine that when you have the amide hydrolysis, this bond breaks down, releasing the fluorophore that gives you like an on signal. So we have this 150 compound libraries that you can screen against sample to identify hidden potential of this sample. For example, you're looking at 2 different conditions where you expect one to have a higher or lower amide hydrolase activity. Then you can screen against this library to identify which condition has higher amide hydrolase function, for example. So this probe library is also available for EMSL users, ready to be used, and already aliquoted into plate readers. So if you already have a plate reader that can do fluorescent characterization, we can actually ship this so that you can do it in your own lab. So the extension of this library is that there is this function, but who are responsible for this function? Which enzymes are doing that, the activity or the turnover? So we brought in the next step where we take the hits from that library and modify it as a activity-based probe that irreversibly binds its target. So once it binds the targets, we can perform pull down protein enrichment and analyze to identify what are the targets that are responsible for this biochemical function. We did similar that experiment in a soil bacteria, and we were able to identify several targets, and those are typically annotated as hydrolases, transferases, in the current the protein IDs. One thing that stood out was there was quite a bit of protein targets that were not labeled or annotated as anything. And with the information that we have now, you can say that those targets likely have amide hydrolase activity. Basically, like you're providing a new experimental information to annotate those proteins. So this is a little different from those probes that we saw. This targets what is called Raman spectroscopy, and these are called Raman chemical probes. This is basically a teaser slide for your next webinar session that Kevin's going to present. Typically, people use Raman spectroscopy for label-free imaging or analysis of their sample. They're looking at their chemical signatures in their sample. Typically, they're rich in these chemical features that is observed in the Raman spectroscopy, and so it is difficult to track those chemical information when it is label-free. And so the sensitivity is also poor. And so the community came up with this Raman tags or vibrational tags. These are these functional groups that have higher sensitivity compared to the typical functional groups that you find in the biological systems. And these functional groups are unique in a way that they show up in the cell silent region of the Raman spectra, so that you can easily track these molecule in your system compared to other signatures that typically show up when you don't have any sort of label. So here again, I have listed 3 different types. The first one is the metabolic labeling probes. These are like a biomolecules that are tagged with these chemical tags. For example, these are already commercially available that you can buy. The glucoside and the fatty acid and amino acid versions and some of the sugar versions. So you can basically incubate these compounds with your organism and see how they take up these compounds by doing Raman microscopic imaging. And the next type is an activity-based probe. This is very similar to the fluorescent probe that I showed you, where the enzyme acts on this compound to release a species that can be characterized using fluorescence. In the previous case, in this case, it's using a Raman microscopy. Here, this falls in the cell silent region that allows us to clearly visualize this under Raman microscopy. And the final probe that we're developing is affinity-based probes. These are targeted at metals particularly, and so we modify the compounds in a way that they selectively bind certain group of metals over the other, and the ligand is coupled with the Raman tag that allows us to differentiate the bound and unbound state of these affinity-based probes. So these can be custom designed for specific question of specific type of metal and particular type of sugar. So that concludes my portion of the talks. Hopefully I've given you enough information so that you are able to suggest chemical probes as one of the analytical tools for your future research and also future proposals. Next, I would like to introduce you to Kevin. Kevin is a staff scientist at PNNL. He's an expert Raman microscopy operator. Kevin.
There we go. Okay. Hello, everybody. Thanks, Sankar. I'm delighted to be here today to talk with you. Sankar gave a really nice overview of the different types of activity-based probes that you can use and the different kinds of information you can get from them. I'm going to be talking to you about and introducing coherent Raman microscopy and showing you how we can use that microscopy in conjunction with these different types of chemical probes to actually visualize the spatial distribution of metabolic activities and track small molecules inside living cells. So this is a little bit different than the examples that Sankar showed, where the cells are incubated with probes and then digested. Here, we're actually going to incubate live cells and look at the spatial distribution of these tag molecules in real space. Okay, so generally, light microscopy concerns itself with discerning an object's structure through its interaction with light. This is a conventional optical microscope that uses a set of lenses and a white light lamp to register an image. Here's an example of that, where we've taken a living cell, something like filamentous cyanobacteria here. And what you can see is that as the light passes through the sample, You can either scatter that light to create amplitude, or the light can interact with phase gradients to create phase images. And generally, we see both amplitude and phase modulations inside these cells. So what if we wanted to get instead composition information? That is, we wanted each pixel in this image to tell us a little bit about what lives there, whether it's lipid, a DNA, or an extrinsic Raman probe molecule. Well, for that, we need to turn to coherent Raman microscopy. Coherent Raman microscopy is a bit different than some of the microscopies that you may be familiar with, including transmission bright field microscopy or fluorescence, which all fall under the umbrella of linear spectroscopies. That is, they involve the linear interaction of light and matter. Coherent Raman allows us to boost the Raman signal using nonlinear effects, which I'll talk about. So what is the Raman effect? The Raman effect is where we take a pump photon and we apply that pump photon to a molecule. And what you can observe is that that light can interact with different normal vibrational modes of molecules. And in the emission, we see Rayleigh scattering, that is light that hasn't changed its energy. We can also see longer wavelength shifted photons that correspond to Raman scattering. Now, interestingly, the way this works is the energy difference between the Raman scattered photons that have excited molecular vibrations and the pump laser, it corresponds to the energy of those vibrational transitions. So when we look at Raman spectra, we're able to discern amplitudes corresponding to different vibrational modes in the system, and that's how we extract composition information. Now, as it turns out, the linear incoherent Raman effect is extremely weak. It's about 15 orders of magnitude weaker than a typical fluorescence cross-section. So in order to exploit it, we need to turn to the nonlinear implementations of that effect through spectroscopies called stimulated Raman scattering or coherent anti-Stokes Raman scattering. Now, unlike the linear variety, where we've applied a pump photon and then waited for red-shifted Stokes photons to come out, we apply multiple pulse laser beams simultaneously to drive those transitions. And by doing so, we can amplify the emission rates of the Raman photons by about 1 billion-fold. And that allows Raman effect to be exploitable for live cell imaging. Now, stimulated Raman scattering and coherent anti-Stokes Raman scattering are two different ways to visualize the same process, where in effect, we're looking at the vibrational features of a specimen. Stimulated Raman scattering is detected as an intensity of the laser source, whereas coherent anti-Stokes Raman scattering arises because you're looking for photons of a certain color. But they're very similar, two different spectroscopies that, in effect, look at the same process. Now, we need ultra-fast laser technology in order to create and synthesize these different pulse laser sources. Our system is a synchronously pumped optical parametric oscillator, and it allows us to generate a 1060 nanometer near infrared pump light and some energy tunable Stokes radiation. And by applying all those photons together in a conventional microscope, where instead of using a lamp, we've replaced that with this laser source, we can actually execute these spectroscopies. Now, what does that look like in a microscope? Typically, we take that incident pulse train of radiation, apply it to raster scanning mirrors, separate it from emission with a dichroic mirror, and then use conventional optical microscopes, photomultiplying tube amplifiers for visualizing it, or actually doing spectroscopy. And all these things are possible. Now, the beautiful part about coherent Raman microscopy is evident in this example, where I've simply taken a drop of olive oil and put it on a cover slip, and then performed a Raman spectrum measurement in the lipid region. And what you can see is when I tune the lasers to 2805 wave numbers, I don't see a signal at all. But if I move over to 3125 or 2846, I see various amplitude signals where I can actually visualize the droplet itself and the boundary where there's just cover glass. Now, there are slight differences between how these images are registered, whether it's the coherent Raman channel or the stimulated Raman channel. I'm not going to get into that today, but the point is each pixel corresponds to vibrational information. If you pick one pixel and instead of looking at an image, look at spectra, here are the corresponding spectra. And what you can see by looking at these curves is that this sample is composed primarily of lipids, which is no surprise. So there are some important aspects of stimulated Raman scattering that are important. It's label-free. We do not have to intrinsically label if we don't want to. It gives us molecular specificity. It allows us to visualize live cells with video rate speeds, and it's not nominally destructive to live cells. So I will give you a brief example of how we can visualize intrinsic or native vibrational signatures in filamentous cyanobacteria. Here's an example of the same measurement looking at Anabaena. And what we can see is in the fingerprint vibrational region, we see a few vibrational features. Now, by comparing these features to conventional Raman spectra of neat protein pigment complexes that we think are inside, we can actually identify these molecules. So one of them corresponds to a protein pigment complex in the phycobilosome. The other corresponds to carotenoids that are part of the photosynthetic apparatus of the system. But the point here is that we're able to see them in real space by looking at the cells. Now, activity-based probe methods are a bit different. Instead of looking at the native signatures, what we're really trying to do is take a molecule that's going to interact with our cells in some way and track it and its spatial distribution. In order to do that, we've been working with several prototypes that involve alkyne or diene chemical functional groups, which, as Sankar pointed out, give us narrow vibrational bands that we can visualize in the cell silent region that doesn't overlap with all of the DNA and amides that you see in the vibrational fingerprint or any of the lipids you see in the high frequency spectral range. So we've been working on using these chemical moieties as different functional groups for preparing a catalog of complexes that we can then use to monitor different processes in different types of cells and cellular environments. Now, on the engineering aspect, one important part is it's important to have as minimal probe volume as we possibly need to avoid cellular toxicity and in order to avoid the probe molecule itself introducing artifacts or changing the intrinsic cellular metabolism. So in order to do that, we're very interested in boosting the Raman intensity of these molecules. And in this measurement, we can see by changing the concentration and measuring the amplitude of each of these vibrational peaks, we can see that two of them, this diphenyl alkyne and this diphenyldiene, actually are visible in the sub-millimolar range in a detectable range for our sensitivity of our instrument, which is important. It's also interesting to note that simply by adding a phenyl group, we were able to boost the signal by a factor of 5 compared to the single phenyl group. And by adding a diene with a biphenyl moiety, we can actually boost it another factor of 5. So engineering these is extremely important for achieving low toxicity and a high enough amplitude to be able to track these molecules in live cells. Okay, and what I would like to leave you with is a very interesting example that kind of incorporates all the different aspects of activity-based chemical imaging with Raman and coherent Raman. Now, this is an example where two different dye scaffolding molecules that contain isotopically labeled nitrile moieties are actually used to selectively bind to copper-1 or copper-2, which then selectively bind to various free proteins inside yeast cells. Now, here, the nitrile groups, the two different nitrile groups, give you a way to image these dye scaffolds inside live cells and to be able to distinguish them based on their isotopically isotope labeled Raman shift. So carbon 14 shows up at a slightly different frequency than carbon 15. And those are used to tell us where those are in space. And then there's this azide chemistry that allows different oxidation states of copper to bind to the scaffold, which then selectively bind to different proteins. So all in all, we're able to visualize different proteins and their spatial distribution all at once in this multiplexed way. So I like this example because it contains all of the elements of all the elements we've discussed: high brightness Raman probe, metabolic labeling, selective protein affinities, and stimulated Raman to allow us to visualize these in living cells. So I left a little table here for you to explore, which talks about the applications of different probe types, whether it be activity, affinity, or metabolic, and a couple of references where you can look up some of those examples. With that, I will turn it over to Damon Leach, who's a data scientist here at PNNL.
Great. Thanks, Kevin. And yeah, my name is Damon, and I am a data scientist and biostatistician here at PNNL. And I am excited to talk with everyone here about like one way that we can process activity-based probe data that we've developed here at PNNL, which is this program that we've called ChemoProt-R. And so ChemoProt-R is an R package that was developed using an EMSL S&T project led by Vivian Lin, and it helps analyze and create reports for both labeled and label-free mass spectrometry-based proteomics data. So when it comes to ChemoProt-R, there are 6 main steps that are going to occur throughout this pipeline. The first is that we're going to run through general pre-processing steps, things like filtering out specific peptides. We're going to run some outlier detections to determine if any samples are behaving differently than others. We'll then run protein rollup, taking the peptide level data and converting it to protein level. Then running statistical analyses such as ANOVA and the G-test. And then finally, once we've ran those statistical analyses, we can then create both a report as well as a results file that the users can then use and kind of dive deeper into if they have any questions pertaining to kind of what specific proteins were doing what with regards to fold changes. But first, and I'll just briefly touch on this, is the input data. This is going to be an Excel file that has multiple tabs, and this is going to vary just a little bit depending on if you're working with labeled or label-free data. In general, though, what we're going to need is we're going to need to have tabs that are including things such as spectral probability values that will be used for filtering, the intensity values of the specific samples that are being done, information about the jobs, as well as like protein collection information, and also a tab that pertains to what we call the F data, which is sample information, basically specifying which of the samples are perhaps the probe samples, which are the no probe, things like that. And if you have any questions about kind of what the structure of these Excel sheets should look like, I highly encourage looking at that repository that we were just talking about, and there should be a link for, because there are going to be examples in that repository from example data sets for both labeled and label-free that can give a better insight of kind of what the actual structure of those Excel sheets look like. But once we have the input data readily available, then it comes to the steps of running through the first of the 6 steps, which was the general pre-processing. And this is going to be done at the peptide level. So with labeled data, we're going to have an additional first step, which is running isobaric normalization based off of the reference samples. But then after that, the steps between labeled and label-free are going to be quite similar. We're going to filter out molecules based off of the desired spectral probability value. Any peptide redundancies are going to be summed up. So if a peptide is mentioned multiple times, we'll sum that into a singular value for each sample. And then also identifying the number of missing values by sample. And that's what this figure here on the right is demonstrating. Next, we can look at potential outliers. Within ChemoProt-R, we use what is known as a robust Mahalanobis distance, and this is going to calculate a score for each sample. And the higher the score, the more likely it is to be a potential outlier. And it uses a couple of different metrics. For this specific project, we use the median absolute deviance, skewness, and correlation. And what we see here is that this fifth sample is a potential outlier based off of the p-value threshold and the log2 distance. But we can do a bit more exploration to kind of how that outlier was determined. And what we see is it has a high median absolute deviance. It's pretty on line with the skewness, and it's a little bit low, but not aggressively low when it comes to the correlation. When it comes to deciding whether or not to remove an outlier, it is heavily subjective. Because we're working with so few samples in this experiment, we may decide to retain it into the experiment just so that way we have 3 from both the probe and no probe samples. And so once we've done the pre-processing, we've looked at outliers, the next step is to convert our data from peptide level to protein level. And ChemoPro supports 3 different types of rollup. That's going to be standard rollup, reference rollup, and summation. And I'll just briefly give an example of each. So if we're doing standard rollup, this is going to take the mean or median of all peptides that map to a given protein, setting that as the relative protein abundance. And so if all 3 of these peptides, A, B, and C, map to a single protein, for sample 1, we'll take the median, which in this case is 17.3. For sample 2, we'll take the median of 18.9 and 17.8, which is 18.35, and so forth. And that's how we're going to get our protein level abundance when we use a standard rollup. However, we also can use a reference rollup, and this is perhaps the most complex algorithm of the 3. And that first, we select the reference peptide, which is going to be the peptide that has the most observed values across all samples. In this case, that's peptide A, because it has 5 observed values out of 6. We then use that as the reference and kind of center everything based off of that. So for example, peptide B in sample 1 is 20.1, which is 2.8 units higher, so a negative 2.8. Whereas peptide C is 2.1 units lower, as it's at 15.2. So we can do that for every single peptide, for every single sample, and get then the median disparity between each peptide from peptide A. So peptide B is negative 2.75 and peptide C is 1.1. We then use that to scale our data to put it on a more comparable scale. And then we do what we did with the standard rollup, which is to take the median. So in this case, it's still 17.3, 18.9, and so forth. And you don't have to memorize this. This is just something that is going on behind the scenes, but it's just helpful to kind of know what the differences between them all are. And then the last one is going to be summation. And summation is pretty simple in theory. You just kind of sum up all of the different peptide values, and that is your abundance. Just the keynote is that we do have to convert our values. They're currently on the log2 scale. We convert them to the raw abundance, sum them up, and then we can then put back onto the log scale to get the final log2 abundance using summation as the rollup. And so now that the data is on the protein level, we've ran through all of the processing steps, we can run our statistical analyses. And so we can run both quantitative analyses such as ANOVA and qualitative ones such as the G-test. And the G-test is going to be based on the number of observed samples that are in each group. So basically, if we have 3 observed for protein A in the ABP samples, but 0 in the no probe samples, that is likely going to turn up as a significant difference. And so this plot just shows kind of some of the different comparisons we can make. Anything that's in green is considered a positive significant fold change, and anything that's red is going to be a negative significant fold change. This first column is for ANOVA for that quantitative result. The second one, G-test, is for the qualitative. And then this third column is going to be looking at both the qualitative and quantitative results together. So this 763 is simply that 558 plus 205. And we can also just visualize it another way. This is going to be specifically for the ANOVA results. We can't really plot a volcano plot for the G-test, but we're kind of seeing the exact same results as we saw on that previous slide. And so finally, once we have the analyses done, we can also create both an Excel file and an HTML report summarizing what we've done. And so the results file will have 2 tabs. It's going to contain information for both the ANOVA and G-test results for every molecule. So it will give us the p-value, whether or not it was statistically significant and in what direction. And then the second tab will then have our log2 normalized abundance values that were used to then process those statistical results. And then we'll have a report that goes through every single one of those steps, showing the pre-processing, the outlier detection, as well as what we did to run the statistics. And with that said, thank you for your time. And if you have any questions about ChemoProT-R, please do not hesitate to reach out.
Thank you, Damon. And with that, we are going to start our Q&A. So thank you for all the folks who added their questions to the chat. We have quite a few to go over, so this is fantastic. Okay, let's start with Fabian has asked, do these probes applied in cultures affect the growth of microbes because of the irreversibility binding probes? Now, I believe that goes to Sankar.
Yeah, that's a very good question. So what we do is usually when we label, we label it in like PBS without any carbon source. These are not culture media, but just the PBS. And you can think of them as a snapshot of the proteins. So you're not incubating the probes with the culture for a long time. And so you're basically taking a snapshot of when you incubate the probe with the target organisms.
Okay. Kevin, so Detta is asking, what other metals can you use? Iron, calcium, are they possible?
Yeah, there's actually quite a few activity-based probes that are based on heme and iron and liable iron pool analysis in live cells. I think the limit here is really your imagination with the type of scaffold that your Raman tag is living on, and whether or not that can be engineered so that it's not phototoxic and it's very bright. But calcium and iron are certainly possible, and there's quite a few iron and iron redox examples. Good question.
So Damon, Fabian is also asking you, why use the median instead of the mean?
Yeah, so ChemoProT-R actually can use both median and mean. That would just be a slight distinction that you make in the code to kind of decide which one you'd rather use. Personally, I tend to, when in doubt, use median just because it is a bit more robust when it comes to kind of like the skewness of the data. But yeah, when it comes to roll up and things like that, you could use mean as well.
Great. Sankar, Yuting Wu is asking, what are the targets of your probes? How generalizable are these probes in terms of adapting to other targets?
All right, that's a good question. So the targets are probe dependent. We design a probe particularly if we are targeting one specific class of enzyme. So we develop in a way that is the only class of enzyme that is targeted. And there is also a narrow spectrum and a broad spectrum probes. The broad spectrum probes kind of targets the whole family of that particular class of enzyme. So the first part of your question, it depends on the probe that we're looking at. So the target can be anything that we like. Remind me the second portion of the question again, sorry.
Yeah, it was how generalizable are these probes in terms of adapting to other targets?
Right. So yeah, I think if the probe is made for a particular class of enzyme for one organism, they are usually adaptable to another organism, but we always do some preliminary experiment to see whether if there is a limitation or is the enzyme class is actually accepting them as a probe or not. So there are certain classes where they are considered broad spectrum, they are promiscuous, and so they do, you can use them across the organism. But if we made a probe that is very selective for one particular enzyme, then it may not be suitable for other organism.
Okay. Kevin, So Anonymous has asked, so Sankar showed data using fluorescent activity-based probes and live cell imaging to look at spatio-temporal distribution of proteins of interest on a conventional fluorescent microscope. What do Raman probes offer beyond fluorescent probes?
That's a fantastic question. So fluorescent intensities are usually very broadly distributed. So the fluorescence emission comes at over many wavelengths, which are difficult to multiplex and sometimes difficult to make chemically selective. The Raman picture is very narrow frequencies, which are completely isolated from all the other features that you'd see in the spectrum. So they're very chemically selective, and they give you this composition information. So when you have the probe engineered so that there's a change in amplitude or frequency or something like that for the Raman probe, it's extremely sensitive compared to fluorescence.
Great. Okay. So Damon, so Fabian is asking, what is the real meaning of finding outliers in this analysis, and what implications do they bring to this analysis?
Yeah, so like for the kind of toy example that I was showing there, when you only have 6 samples, looking for outliers is probably not the most helpful, just because removing one sample is such a significant part of your data set. But if you're working with maybe like a system that has like an 18-plex or something, where maybe you have like 9 samples per, like for the probe and 9 for the no probe or something, running that outlier analysis and finding like a bad sample, which then leaves you with like 9 and 8, is not nearly as extreme. And so if we keep outliers in the data set, it could skew some of our results, especially if we know that maybe there was a reason why that specific sample, maybe it was like processed incorrectly or something like that, where we can kind of use this outlier analysis to help quantify what does and doesn't make sense to kind of retain in the data set. But yeah, when you have so few samples, it kind of doesn't make sense to really remove them.
Okay. And also of note, so Damon, the link that was provided to everyone to access, I believe it was the link code. I think it was to access your chemoprot. It looks like you have to be an active user in order to access the link. So I just wanted to let the audience know, because we've had a couple folks mention in the chat that the link didn't work for them, and that's because you have to be an active user to access the software, which is more incentive to get your proposals in. So next question for Sankar. When you identify the unknown proteins using your probe, what is the limit of sensitivity? If I understand the technique correctly, the technique only works if the enzymes just bind, not catalyze the probe. Is that correct? And what is the probability of off targets based on your experiences? Happy to repeat any of those parts of the question.
Okay, so the first question, the sensitivity depends on the probe. There are cases we usually, when we develop a probe, we perform what is called a dose-dependent labeling. So we identify what is the minimum amount of probe you need to characterize that particular targets that we're looking at. So the answer is it's probe dependent, and there are really very sensitive probes out there that can detect a really small quantity of the targets. The second portion of the question, that is correct. So there are the probe themselves are different way we can develop. There are cases where the probe acts as a covalent inhibitor, basically it shuts down the enzyme. And there are cases where the enzyme acts on the probe and turns over the probe and binds the proximity, not necessarily the residues involved in the catalytic activity. So that even after binding the enzyme still may be active in those cases. And the last portion of the question.
What is
So-
the probability of off targets?
Right. So yeah, I think there's definitely off targets, and it's really difficult. And it involves a lot of background work to come up with something that does not have any off target. Particularly the photo affinity probes do tend to have off targets, and they are promiscuous. And so what we do basically to minimize that is to increase the number of replicates, one, and in cases where we have access to the substrate which we're using to mimic with the probe, we perform what is called competitive protein profiling, where we are first treating the probe samples with the actual substrate and then treat it with our probe to see if they're binding the same location. So that also helps you further narrow down and filter out the off-target labels.
Okay. Kevin, Uteen is asking, how do you track the real-time distribution with irreversible bind sensors? And does the Raman microscope need to be adjusted for each sensor, how the signal was covered to picture, and the resolution?
Okay.
How-
Yeah, so if the chemical process is irreversible, you would need to make sure that the frame rate of the instrument is faster than that chemical process in order to visualize it from start to finish. Remember that not all of the probes that we use involve metabolism of the Raman probe itself. So even though different probes can undergo different chemical processes, you can still see the Raman signal persist as long as it's inside the cell and it hasn't been digested or metabolized. The next part of it is, yes, so depending on the type of chemical probe you're using, the instrument will need to be adjusted for that. It also will need to be adjusted for different size cells, depending on what kind of populations you want to look at. Typically, what you would do is take the chemical probe itself without incubating it inside the cells and characterize it with sensitivity and obviously get some spectra. Then you would find out, okay, now that I have a Raman frequency of interest, I can do live cell imaging at that frequency. If the different molecules have different vibrational modes, and that's what you would need to change in order to visualize different probes. The resolution here is generally diffraction limited. So imaging with coherent Raman typically takes place with the laser sources out in the near infrared. And this is a pretty non-perturbative region for live cells, so that's helpful. But that also means, the diffraction limit is, roughly the wavelength over 2, so we're talking about hundreds of nanometers. Stimulated Raman and coherent Raman can be executed in the visible and UV, but the laser sources get much more destructive as they get higher in energy. So that's generally a technical limitation that's being addressed with other bioimaging techniques.
Great. Okay. We are at 1 o'clock. So right on the dot, want to thank our presenters for your fabulous information today about activity-based probes, Raman spectroscopy, and ChemProt-R. Thank you everyone for attending. Hope you have a great rest of your day. And we will see you on April 9th for our next EMSL LEARN webinar focused on the P-Flow-Trans subsurface transport modeling software. Thank you, everyone.